Good day, ladies and gentlemen, and welcome to the Arthritis Foundation Taking Control of Your Arthritis conference call. All lines have been placed on a listen-only mode, and the floor will be open for your questions and comments following the presentation. If you should require assistance during the conference, please press *0 to reach a live operator. Unfortunately, Dr. Harden will not be joining us today, but we are pleased to announce we have a special guest. At this time, it is my pleasure to turn the floor over to your host for today, Dr. Tuan. Sir, the floor is yours.
Hi, everybody. Welcome to this event. I'm really delighted to have the opportunity to speak to all of you. Just a short introduction of who I am. I am a professor in the University of Pittsburgh School of Medicine. I direct the Laboratory of Cellular and Molecular Engineering, and I also hold a chair endowed chair in sports medicine. So my own personal research expertise is in the field of tissue engineering and regenerative medicine, specifically using stem cells, smart biomaterials, and molecular and genetic techniques to develop new therapeutic approaches for skeletal diseases, particularly osteoarthritis, which is the topic I'm going to speak on today. Again, welcome all of you, and I know you all have a lot of desire to find out more about the disease and what can be done and so forth and so on, and I'll try to do my best to address some of these in my talk, and then, of course, I will be very happy to take questions afterwards. So my research focus, as I said, is on osteoarthritis, and specifically there are three topics I want to address. The first one is what causes the disease, the breakdown of this joint that normally works very well for all of us. And then secondly, what can be done to halt or to stop this degeneration? And then thirdly, how can we treat and repair the degenerated joint? All right, to start off, let me just say something about osteoarthritis. What is osteoarthritis? The easiest thing is to look at the name. The name has osteo and arthro and then itis. Osteo means bone. Arthro means the joint. And then itis is a disease. So it's really a disease that involves the joint. And we have, of course, many joints. They're usually referred to as articulating joints because they help us move. And the disease generally involves the degeneration of two main tissues. The first one is cartilage. The second one is bone, and that's why the osteoarthritis name. What is this joint? Well, the joint is actually very similar to, say, a ball joint in a car. So where two pieces are meet up, and they're able to move with respect to each other, and they're usually contained inside. In the case of a car, it will be a boot that is like a shell that wraps around this joint. And same thing for our human joints. So the two joints that are mostly, I guess, the biggest target for osteoarthritis would be the hip and the knee. And in the case of the knee, you have your thigh bone and your shin bone, the femur and the tibia meeting each other. And at the end of this two pieces of bone, there is a piece of Teflon-like tissue called the articular cartilage. I'll just refer to it as cartilage. That is very smooth and allows the joint to really move very easily with respect to each other. And, of course, inside this capsule, which is called the synovium, you have the fluid in there. So basically, we are literally walking on water because the joints actually never touch each other, and they're sitting next to each other, but there's a bunch of fluid surrounding this. Now, what happens when you have osteoarthritis? Well, what happens first is that usually the cartilage degrades. There are little fractures showing up in that cartilage, and then eventually all kinds of tissue reactions happen in this joint. such as the bone underneath starts to also degrade, And then you have things like bone spurs showing up as a reaction from the bone. So you have little pieces of bone sticking out. And you have inflammation that gives you the swelling and redness, tenderness. And then the best way, or in fact the only accepted way to detect osteoarthritis is by imaging. Using X-ray or MRI, you start seeing that the gap that normally exists between the two bones, because as I said before, we walk on water, that gap narrows. It gets smaller and smaller. So now, if the cartilage now goes away, what you have is bone on bone, and you can imagine that that hurts. And then finally, the most important and recognizable signal is that it hurts. All right, so pain is, of course, associated with this, and at some point you also lose mobility and so forth and so on. Now, if you were to take a look inside the joint, arthroscopy is the method that is used. If you were able to take a little camera and go inside the joint and look at this point, you will see that the surface cartilage is flaking off. Right? It's like paint coming off from the wall. And so as a result, this Teflon coating, this protective coating is no longer there. And so that's when you have full-blown osteoarthritis. So that's the disease. So what kind of treatments are currently available? Let me start off by first saying that there are no osteoarthritis drugs, disease-modifying drugs of osteoarthritis available right now. That's just the way it is. So what do we do then? Well, there are basically three main methods, three main approaches that we take to address all these symptoms associated with the disease. I will just lump them into pharmaceuticals, nutraceuticals, and lubricants, basically. So let's start with pharmaceuticals. Pharmaceuticals, I think most of you are probably familiar with the non-steroidal anti-inflammatory drugs, generally referred to as NSAIDs. And they are basically painkillers. They are anti-inflammatory, so cuts down the swelling, cuts down the soreness, and so on. Nutraceuticals, and of course there are many of them on the market, glucosamine, chondroitin sulfates, MSM, fish oil, etc., etc. And then finally the lubriceuticals. Essentially, at the end, as I told you before, what you have is you have a joint that is not very well lubricated anymore. It's stiff. And so what you do is you inject things that are normally found in the joint fluid. A most commonly used one is hyaluronan. It's a big name for also a very big molecule that you inject there. It's basically a lubricant, right? So you do a lube job. So pharmaceuticals, nutraceuticals, and lubriceuticals. None of these, as I mentioned already, really cure the disease. They are more treating the side effects of the disease, right? Sometimes they're effective, sometimes they're less than effective. So what else can we do? Well, in the early stages, there are, of the disease, there are ways that you can move a piece of good cartilage into the place where there is bad cartilage. Okay, so I usually refer to this essentially as the hair plug transplant approach. You are a little thin on the top, you will move something from the bottom to the top. And you can immediately see that this is basically a zero-sum game. You are not making any new hair, you're just moving hair around. In the case of these cartilage transplantation, the same problem exists. You're not making any new cartilage, you're just moving cartilage around, and the place where you get that good cartilage may eventually actually also develop osteoarthritis. So, again, most of the drugs are anti-inflammatory, just trying to block the body's response to disease, and there are no drugs available on the market that actually work on the target tissue, which is, first of all, cartilage and also bone. Now, I think also most of you are also familiar with total joint replacement. That is the ultimate irreversible solution. What you do then is you remove the biological joint and you come in with artificial materials, mostly metal and plastic, and you insert this into the two pieces of bone that make up the joint, and now you allow this material to replace your joint. Well, what's wrong with that? Well, there's actually nothing wrong with that because these materials don't ever go away, which is good. They will never degrade in the way that our natural material, natural tissue degrades. The problem is not the material. The problem is the interface. Because remember, this thing goes in and it's sitting somewhere where there used to be nothing like that before. So it has to interface. It has to bond with the original tissue. And that's the weakest link of the total joint replacement approach. It works very well, but eventually that interface will fall apart. The general life expectancy is about 10 to 15 years, sometimes shorter, but very rarely longer. It is okay for individuals who are at a certain age where that is actually a significant number of years, but unfortunately as the population that requires total joint replacement decreases to, say, 40-year-old, well, the 10, 15 years is no longer workable. So oftentimes you end up with a revision, and as you can imagine, when you quote-unquote revise, meaning you remove the original one, you have to dig a bigger hole. And obviously there's a limit to how big a hole you can dig. So I think you can see the limitation there. So therefore, we got to do something. Now, the big problem right now is that we don't know that we have osteoarthritis until the pain comes in. By that time, you're already pretty far down on the road to irreversible degradation. So it would be very nice if we can actually find out when osteoarthritis begins so that we can do something early. Obviously, it would be even better if we can prevent this from ever happening in the first place. So to better illustrate this, I would like to use the pothole on the highway or on the road as a good example. Now, what we have right now for osteoarthritis is that our car just got smashed because we ran into a pothole. We realized there was a pothole. So how did this pothole ever come about in the first place? Well, there are a couple of possibilities. The first one is, well, it was poor workmanship from the beginning. Poor materials, very bad asphalt material put in there. And so I would equate this to genetic origin of the disease, right? Remember, the pothole is now the equivalent of a degraded cartilage in an osteoarthritic joint. Well, it could be genetic, meaning you start out bad already, although initially you can't tell, but because you're starting bad, so eventually it goes. All right, so that's one possibility. The second one is wear and tear. Well, you're on I-95 or whatever, and a lot of cars going by every day, heavy traffic. Well, that could be the reason also. And then the third possibility is environmental. It could be that the weather is bad and salt comes in and so on and so forth. So that's equivalent to bad nutrition, bad lifestyle, et cetera, et cetera. And then the final thing is trauma. If there ever was a very heavy load dropped onto the road surface, or there was a traffic accident, there was a fire, well, that's trauma to the road surface, and as a result, you end up with a pothole. Now, of these causes, the genetic causes, the wear and tear, the environmental reasons, or trauma, only trauma is the one possible cause that we actually know the origin. Trauma is, well, in the case of the road, we say, well, there was a car accident, and this weight got dropped onto the road, or there was a fire. We know starting from that day, there probably will be a pothole later on because of this trauma. So likewise, for us, human beings, trauma such as, say, sports-related trauma like ACL injury, meniscal tear, blunt trauma to the knee as a result of a car accident when your knee hits the bottom of the dashboard, those things we can actually identify and say, well, that's maybe the beginning of all these bad things later on. And in fact, the studies have shown that approximately 15% of osteoarthritis can be traced to a particular traumatic experience, right? But again, it's not everything. It's only 15%. But that's the one that we have some idea that can lead to the development of osteoarthritis. So that's why we have seat belts. That's why we have all kinds of protective gears for the sports players, and also good training to make sure that the weight bearing is right, and so on and so forth. So that's kind of this is all we know about the cause of osteoarthritis. As I said before, the environmental causes like weather or wear and tear due to aging, due to heavy use, and so forth and so on. With all those things is that the problem is we still don't know when it starts. So it's very difficult to figure out when to start looking at treatment. So what can be done? Well, as I said before, timing is critical. If we can find out whether there's say due to trauma, we know, let's say osteoarthritis, the disease actually has its beginning 2 days after the trauma. Well, then we can do something maybe within the first 48 hours to stop this process from ever happening in the first place. So timing is critical, and in fact, some of the treatments, such as particularly the nutraceuticals, the glucosamines, the chondroitin sulfate, and so forth and so on, there's some evidence to show that if you can have this type of treatment, most of them, by the way, are selected because they are the precursors to the material that will be deposited into cartilage later on. So we figure if you have more of the goodies, then maybe the cartilage will be a little healthier. There's evidence to show that if you do that early, let's say after trauma, then the results may be a little bit more satisfactory. So again, but the verdict is still not in, and we need to kind of await more data. Now, as I said before, unfortunately, usually one does not find out that one has osteoarthritis until the tissue is already damaged. So therefore, what can we do? We can, number one, stop the degradation process. Okay, the bad things have already started, but if we can stop it, no more, that would be nice. Well, degradation generally is carried out by enzymes that are degrading enzymes that will chew up the protein, chew up all the matrix that's in the cartilage. So we can find an inhibitor to these enzymes. That would be nice. A lot of inflammation results from the falling apart of the tissue. If we can stop the inflammation, perhaps we will also stop the degradation process. Another way, a little even bolder approach would be, can we reverse the process? So things have already fallen apart. Can we actually stop it and then build it back up? That is very difficult for a very simple reason. Cartilage is a tissue that we have made for the purpose of it staying around for a long time. In fact, it has been shown that for someone, let's say in the 50s or 60s, most of the molecules in your cartilage tissue, in the joint, were actually made when you were a teenager, right? They never go away. And it's very important that it stays that way, because we don't actually want cartilage to become mushy at any time. We want it to be around for a long time. It has its downside. The downside is that the cells that make all this stuff in the first place, they don't make this anymore because they don't need to make it anymore. And then so when something falls apart, if you want to wake them up, is not that easy because they're not used to making new things. So reversing it is very hard. So this is the challenge right now, stopping or reversing. These are very difficult problems. Now, you may say, well, that's why we have pharmaceutical companies. That's their job, to develop drugs that can do this. Well, the number of companies that are working on osteoarthritis drugs is actually diminishing. In the 1980s, there was a flourish of activities. Many pharmaceutical companies get involved for a very simple reason. Up to 25% of any population has degenerative joint diseases, and most of that is osteoarthritis. That's a lot of people, and therefore, from the vantage point of the pharmaceutical companies, potentially a lot of revenue. So it is a no-brainer to get involved. Well, why don't they stay there? Well, the problem is it's very difficult to develop this drug. As I said before, you don't realize it until it's a little late. And secondly, how do you test this drug? Because you can only test on animals or people, perhaps, hopefully, at an earlier stage, but you can't find those people. And so as a result, they have not been able, no one has succeeded in developing a disease-modifying osteoarthritis drug. And generally speaking, in the pharmaceutical industry, only 1 out of every 10,000 laboratory-developed or laboratory-discovered compounds ever becomes successfully launched. So each launch, therefore, can cost up to $5 billion. And the failure usually is not in the laboratory. The laboratory actually works very well. You identify something as a potential candidate, you take it to the animal level, and then you take it finally to the human level, and that's where the failure happens. It's very expensive failure because these are expensive studies. Sometimes it's because eventually it becomes not useful because it turns out to be not very effective. And most of the time, however, it's because it's not safe. It has other problems. It can cause cardiovascular problems. It can cause inflammatory problems, and so on and so forth. It's very difficult. So what we really need for this field is a platform that can do this right, meaning it can represent multiple tissues and can develop a model which allows early assessment. And so that's where things are at this point. All right, so now I'm going to tell you a little bit about the promises that are in front of us. So treatment and repair. A lot of the stuff going on in my laboratory utilize stem cells and smart material as a platform to make things that can be used to repair or resurface that articular cartilage. The cells that we use are stem cells. Now, these are adult stem cells, meaning they can be taken, in fact, from the patient himself or herself, generally from either bone marrow or fat tissues, from liposuction as a source. The materials, the requirement here is as follows. We need to make something that can fit into that very irregularly shaped a lesion of cartilage. It has to be compatible with the rest. It can integrate very nicely into all the nooks and crannies. And ideally, it should go away when the job is done. It should be biodegradable and not like plastic and metal, which is in total joints. The two approaches that are available right now is, first of all, make this outside, assemble it outside, just like a total joint, except you don't use plastic and metal. You use cells, you use these materials that are degradable, and you make one exactly as the tissue will require, and then you surgically implant this. That's one approach. The other one is to assemble it inside at the site. And so that's the one area that we've been actually making a lot of progress recently. We've been doing a lot of stuff outside as well, but for the assembly inside, we use 3D printing, and we use sort of a photo cross-linking approach to take the cells and the material, the biomaterials, and assemble it right there and then. Essentially, the procedure simply described would be to inject something into the site, spread it around, get everything filled in, all the nooks and crannies, and then shine some light, and then let everything polymerize and set in, like making Jell-O, in place. So now these things are actively being pursued. There are many barriers still. The barriers are a lot of times you need multiple surgeries because you need to get the cells first, and then you have to then you grow them up, and then so that you have enough of them, and then you have to do the next surgery, which is to do the implantation. And loading the cell into this material is non-trivial. So these are the barriers. One more barrier, and this is one that is a very big one, is that everything that one does has eventually to receive approval from the FDA, Food and Drug Administration. And the Food and Drug Administration right now has very explicitly stated that the only cells that can go back are what is called minimally manipulated cells. Right? This is actually quite well defined, and so if you were to take cells and you go put them into a culture and have more of them, that's not minimally manipulated anymore. So as a result, a lot of these procedures at this moment do not have the blessing of the FDA. So, but things are moving along, and we're hopeful that in the not too distant future, such procedures can be done. So the final, very quick thing I want to say has something to go back to the drug development phase. As I said, the problem at the end is still facing the drug development approach is that we don't have a platform that's easily usable, that's friendly, user-friendly. So we need a platform that's more reliable, more sensitive, ensure safety so that we know about toxicity and systemic reactions as early as possible. It has to have high throughput, and most importantly, has to be based on a human platform. A lot of the studies, drug development, you have to go through animals, and animals generally do not necessarily reproduce faithfully the results that you will see in a human. So using human cells as a platform. So what we have done recently is we've developed a live miniature model of the joint tissues that contain both bone and cartilage. We call this a micro-osteochondral tissue, which is a little cylinder that's less than 0.2 of an inch in diameter and twice as tall. This is constructed using human bone marrow-derived stem cells and then assembled using 3D photo printing technology, with each microtissue capable of living 4 weeks or more in the laboratory. Right? So with this approach, we can make many, many identical units of, quote-unquote, the joint. So we can use this approach to answer several questions. Number one, the one that I mentioned very early on, how does OA get started? Right? Remember I said it's very difficult to pinpoint when OA starts. Well, we can use this system to investigate what exactly are the factors that get this process started. To some extent, this is just like the civil engineers who need to build bridges, roads, dams, etc., and what they always have is a model system in the laboratory, usually a fairly large model, not 0.2 of an inch, to simulate what's going on first before they actually go out and build a bridge, build a dam, and so on and so forth. And so that's one application. The other one is to test drugs. As I mentioned already, test for efficacy because now we have a tissue, and also for safety. Does it hurt the system? And then finally, to assess the systemic effects. So we can assemble, in addition to the joint, other things such as the liver, the heart, and then connect up these microtissues to see if any of the candidate drugs for osteoarthritis will harm these other tissues before we test any of them on people. So I think I will finish up here by saying that I think we all know, us researchers in the osteoarthritis field know what the osteoarthritis community wants. The osteoarthritis community wants to have a cure or a successful treatment of the disease. And working towards this goal is what we want to do, and hopefully we'll get there relatively soon. So that's the end of my talk, and I'll be happy to take questions.